So hello everyone. So I'll be disclosing here that I'm going to teach photosynthesis in higher plants, and all the important points of NCERT I'll cover here because I've gone through so many types of questions of this chapter. So exactly I know which line is very important. So I will be making you aware of all the lines and the question that can be framed. Okay, means whatever I'll say it's important, it means their questions have come from there. Questions are framed from that part of line, that line. So that is important, so you will get an idea. So for in the higher plants, initially there is nothing so important. What we need to focus here is from the early experiment, the scientist part. So total how many scientists? If the best way to remember is remember the sequence in which the scientist is given. First Joseph Priestley, then which scientist. That sequence, if you can remember it, it will be easier to memorize. So first of all, Joseph Priestley and his contribution. You will read this part, and this is easy part. One read, you will get to know everything. So Joseph Priestley and his conclusion. Okay, Joseph Priestley and his contribution, you will come to know here properly. Its conclusion and everything is properly given. He concluded that a burning candle or an animal that breathed the air both somehow damaged the air. I don't think there is need. The first is Joseph Priestley. Then the second scientist is your Jan Ingenhouse, I suppose. Yes, Jan Ingenhouse, as you can see, this is the second scientist. And the third scientist is here. We have Julius von Sack and his contribution. Generally, if you see glucose, you can directly say it's Julius von Sack. That way I should remember, okay, for easier purpose. And T.W. Engelmann is the fourth one. So cladophora is associated with T.W. Engelmann. That way you can remember. And another one, absorption spectrum. That is also important. Absorption spectrum. If we say chlorophyll A and B, that is also associated with T.W. Engelmann. Okay. So this way to remember. And then another we have is Cornelius von Neal. Yeah, Cornelius van Neal. And his contribution, he's associated with purple and green bacteria. Whenever you see, you can easily say that he's associated. And the most important part is here. Here are a few lines which are very important, like this line. Water is the hydrogen donor and is oxidized to O2 in green plants. Okay, this line is important because they will ask you sometime hydrogen donor. Water is a hydrogen donor and is oxidized to what? Then you must be able to say it is oxidized to O2. Okay, there will be questions framed on this. And what if in place of water, if we use H2S instead, hydrogen donor, that is for the purple and green sulfur bacteria, we will see that H2S will be associated with this too. Means here also the hydrogen donor only, the donor is hydrogen only, but we use H2S instead of water in case of purple and green sulfur bacteria, what we see. And in green plants, the hydrogen donor, we take it from water. Are you understanding? That way. And then another line we have, this is also important but not so, you can just go through. Okay, till here you can go through. What is important here is that the Cornelius van Neal, whatever, he said that O2 evolved by the green plant comes from water and not from carbon dioxide. So important. They will keep asking you, okay, this line, question will be framed. O2 evolved by the green plant comes from water. So whatever oxygen is evolved, it is coming from water and not from carbon dioxide. So if question comes, the O2 evolved, is it from carbon dioxide? Then you have to say no. The O2 evolved is coming from water. Okay, this is important. What you can remember is one at a time. That is O2 evolved by the green plants comes from water. So the wrong statement, whenever it will come, you will be able to make out. Okay, that was important, all that. This is here important from this page. This is actually I'm using the new NCERT. By the way, my old NCERT is not like this. So it's fresh enough, not like that. Okay. So now before we go to where does photosynthesis take place, we will give a, here they have said that in chapter 8 you read about chloroplast. Okay, so about this chloroplast. So before I jump directly into this, it's important that once again you revise the concept of chloroplast. I will do that for you. You will see it will become interesting after that. Okay, so let's go back to that part of chapter 8. So yeah, here it is. So if you properly see, here in the plastids section, initially they will talk about all these types and different types, amyloplast, levoplast, all that, their functions and everything. That one I'll not teach. I'll directly come to chloroplast because that what we need to know right now. First of all, we can see the diagrams here. Diagram is also very important. Let me introduce with a diagram. If we take one coin, if we assume one coin, that is thylakoid, and when so many coins are arranged together like a stack, no, so these we are calling it grana or granum, depending on singular or plural it will go on. So thylakoid is one single coin, and when we keep all the coins together we'll call it grana. And these grana they are connected. Can you see there is a connection, and that connection we call it as stroma lamella. This is the basic idea. See the labeling, you'll better understand, isn't it? This is stroma lamella. Can you see? Yes. So you see, always whenever diagrams are given, please go through it. You'll be able to relate the content more well. Okay. So I've already given the introduction of what is thylakoid, what is grana. Now regarding the space, there used to be a confusion regarding the space. See, I'll tell you, if this is the chloroplast, the space inside the chloroplast is known as the stroma, and the space that is inside the thylakoid, that one single coin that I said, no, if we take one single coin, that is thylakoid, all together is grana. So one single coin which we are calling it thylakoid, so the space inside that is called as lumen. That is very important, because in the upcoming part of the chapter we will see that we will talk about lumen. Okay, and when they are talking about lumen, it means they are talking about the things that are going on inside the thylakoid and not inside the chloroplast. But we need to know that thylakoid is inside the chloroplast only, and we are talking about the lumen of the thylakoid. So you must be able to visualize what I am actually talking about. Whenever I say lumen of the thylakoid, means this whole one single coin inside that space. Okay, the space inside the thylakoid, that is known as lumen, and the space inside this entire chloroplast, that is known as stroma. As you can see, this is stroma. Okay, here they didn't show the lumen, but if I tell you frankly, if I make a arrow from here, that is a lumen. Okay, so if we come here regarding the chloroplast, then we will see in green plants, chloroplast is mainly found in the mesophyll cells of the leaves. Okay, regarding the shape, not much questions come. This is important, that how the chloroplast number varies. Okay, like in Chlamydomonas, 1 chloroplast per cell, but in green alga it is 20 to 40 chloroplast per cell in the mesophyll. So that is important. Chloroplast is double membrane bound. There will be coming questions on organelles which are double membrane bound. Then you must, if you see chloroplast, then yes, it is. And there are many other also where it is double membrane bound. But for the timing, we need to know that chloroplast is double membrane bound, where the inner chloroplast membrane is relatively less permeable. From there, so from here, what can we interpret? We can interpret that the outer chloroplast membrane is more permeable. It means if they are saying that inner one is less permeable, then we must ourselves interpret that outer chloroplast is more permeable, because sometimes there will be assertion reason based question that outer chloroplast membrane is more permeable. Then, so this way, if you have, while reading only, if you make all these interpretations, no, during the exam you will save time. Otherwise you will think there was mentioning about inner chloroplast less permeable, oh, then outer chloroplast, it will take time. No, once you interpret it, immediately when you see it in the question paper, you can immediately answer, because time management is so important, okay, in NEET exam. So you'll understand this once you appear for the test. Okay, you'll come to know how each and every second will have its own value. Anyways, so this is about the space which I said. Inside the chloroplast, the space will be stroma, and flattened membranous sac called thylakoid, present as stroma, stack like already I said grana. Intergranal thylakoids also we can say. If you see this new word, sometimes the question paper don't get confused. Generally we will not see using this, but sometimes to trick you they might use this also instead of grana. Intergranal thylakoids, so don't be confused. Okay, stroma lamella connecting the thylakoid. Stroma lamella description is given here as flat membranous tubules. Okay, okay, here again about lumen it is mentioned that membrane of the thylakoid, and thylakoid inside space is lumen. Okay, and inside space of the chloroplast is stroma. So regarding this stroma, there is a mentioning that stroma contains enzymes which are required for synthesis of carbohydrates and proteins. So if they ask you lumen consists of all these or stroma of the chloroplast consists of all these, then you must be able to make out that stroma consists of enzymes required for the synthesis of carbohydrates and proteins. Okay, and it also contains small double-stranded circular DNA molecules and ribosomes. So sometimes you might wonder, is there ribosomes in chloroplast? Yes, there is ribosomes in chloroplast as well, and in chloroplast ribosome type is 70S. The smaller unit is present in the chloroplast. But if you see in the cytoplasmic ribosomes, there is 80S. So ribosomes has its two types, right? If you read this ribosome unit section, you will come to know there are two types, 70S, 80S. 80S is generally we find in eukaryotes and 70S in prokaryotes. But we also need to know that in chloroplast, the unit is 70S. And cytoplasmic ribosomes means the organelles which are present in the cytoplasm of the cell. There, the ribosome that we find is 80S. Okay, like in endoplasmic reticulum, we have ribosomes, isn't it? So there it will be 80S because it is present in the cytoplasm. But all we need to know in chloroplast, which unit is present? 70S. The smaller unit is present. Okay, that much you can remember. This is all about chloroplast. I hope you have got an idea. Remember, this line is very important regarding the stroma of the chloroplast contains enzymes for the synthesis of carbohydrates and proteins, and it also contains small double-stranded circular DNA molecules and ribosomes. So chloroplast it also possesses ribosomes. It also possesses circular DNA, double-stranded. Highlighting part is double-stranded circular DNA. Okay, so where does photosynthesis take place? The answer is in the chloroplast, and we already have gone through the detail about the chloroplast. Here the detailed diagram is more properly given. It is the outer membrane of the chloroplast. This is the stroma lamella. This is the inner membrane. The outer membrane is there, and this is the grana. This is the stroma. Ribosomes are present, as I've already mentioned. What type? 70S type. Then starch granule is there. Lipid droplet will be there. Didn't they mention about thylakoids? By the way, one if you take, it is thylakoid. Altogether, all the thylakoids you take, grana. We can say combination of thylakoids is equal to grana. So in this part, all the detail that was mentioned earlier, that is only given. Maybe it is given in a brief form, but there we got it in a detailed format. So here, what we need to emphasize more is that in chloroplast, two type of reactions will take place. One is the photochemical reactions, which is known as the light reactions, and all the carbon reactions will be known as the dark reactions. Both the reactions are taking place in the chloroplast itself. This is very important because I still remember one time there was a question regarding dark reaction, whether it is taking place in chloroplast or not. I got tricked for a while because I didn't emphasize more. But if you read properly, the interpretation or the meaning will come out that both the reactions are taking place in the chloroplast itself. I got confused. Later on you will get a concept of PS1, PS2. So I got confused seeing that. But it was basic only. Both the reactions are taking place in the chloroplast itself. And in this chloroplast only we will see PS1, PS2 will come across cyclic, non-cyclic photophosphorylation. All these will come in the upcoming chapter. So this much only. There is nothing much more. What we need to remember, that light reaction, the product is NADPH, ATP. These are the products of the light reaction, and the products are going to be utilized in the dark reactions. Okay, whatever product light reaction is going to give, that is going to be used as a reactant in case of dark reaction. Little bit I told it in the form of chemistry. Okay, interesting now. In this, what is important is you can go through different types of chlorophyll A, B, their color accordingly is given. Carotenoids, yellow to yellow orange, xanthophyll yellow. This part I can tell you that match the following type of used to come. Even this time I think match the following type of question came, where chlorophyll A was associated with bright or blue green, how in the chromatogram, what color they will give. So exactly all the four came in one column and their colors in the other. You had to match. So this part is important from here. Remaining in this part, not so. These graphs are very much important because many a time they will draw this graph and they will ask you which is carotenoids. They will label it, no, A, B, C, D, and they will ask which one is carotenoid here, which one is chlorophyll A graph. So you have to very properly see the graph. How to remember is chlorophyll A little bit, okay, but you see chlorophyll B, the highest peak. If you see the highest peak, that is chlorophyll B. Then a little lesser one will be chlorophyll A, and another one which has two peaks of equal height, and that is carotenoids. This is how you can remember. Here there is no xanthophyll, only 3 of them are there. And what is in the y-axis, what is in the x-axis, very important. What is in the x-axis? Absorbance of light by chloroplast pigments. Okay, this is important. And here if I come, they will ask you, which one is the graph for rate of photosynthesis, which one is the graph of absorption. So this is the graph of absorption, this is the rate of photosynthesis. I will remember once this graph got added here, and then this one or this one of the graph was included here itself, and they were asking it. So that way also the combination of graph question may be also asked. This is also something there, but not this. I'm talking about this and this. So properly remember the graph. Okay, so that is all. You can go through this part as well. The first one is showing the absorption spectrum. This is absorption spectra, okay, and this one is action spectra. Absorption spectra and action spectra are different. This is the graph for absorption spectra, this is the graph for action spectra, and this is the superimposition of action spectra and absorption spectra of chlorophyll A. I hope you have got it. Okay, an important conclusion of this graph is, we know that in a wavelength of light in the blue and the red region, the absorption spectra will be maximum. This is what they have written it here also. The photosynthesis takes place in the blue and red regions of the spectrum. Okay, most of the photosynthesis will take place in the blue and the red region of the visible spectra. I hope you know all this at least. EMW, he will get in class 12 EMW. There is a chapter in physics. They will be introduced with the, actually you know it already. I don't think I need to tell. So, and another is, see, in other wavelength of the visible spectrum also photosynthesis will take place, but most of them will take place in the blue and the red region. That is important. And see, when first time I came across this, I used to think what it is. So let me tell you, chlorophyll A, most of us know that this is a pigment, isn't it? Similarly, chlorophyll B, carotenoids and xanthophylls, these are also chlorophyll pigments. Okay, and what are these pigments responsible for? These pigments are responsible for trapping the light. As we know, for photosynthesis we'll be needing light, so they are responsible for trapping the light. All this pigment. So though chlorophyll is a major pigment responsible for trapping light, here chlorophyll means they mean chlorophyll A. They are missing here, but chlorophyll A is the major pigment responsible for trapping light. Other thylakoid pigments like chlorophyll B is there, xanthophyll is there, carotenoids are there. They are known as accessory pigment. So now all the 4 pigments that I have mentioned, chlorophyll A, chlorophyll B, xanthophylls, carotenoids. Now let me divide this into 2 sections. In one side there is chlorophyll A, which is the major pigment which will be responsible for trapping the light. And the remaining pigments, that is chlorophyll B, xanthophylls, carotenoids, they are known as accessory pigments, and they have 2 functions or 2 roles in photosynthesis. One is they are going to absorb wider range of wavelength of incoming light. Okay, they will absorb a wider range of wavelength of incoming light and they will transfer it to chlorophyll A. They will absorb as much as light they can and then they will transfer it to chlorophyll A, which is a major pigment. That is the first role of accessory pigments, okay, of the remaining 3 pigments. First role is that they will absorb light and transfer it to chlorophyll A. And the second role is that they will protect chlorophyll A from photooxidation. They will protect chlorophyll A from photooxidation. So these are the 2 roles of accessory pigments, that is chlorophyll B, xanthophylls, carotenoids. Okay, that is all you need to know. That is important from here. Now regarding what is light reaction and the rest of the part, I'll cover in the next video, because it's not possible otherwise while uploading it will take a lot of time, and even the quality content I need to keep up everything. So we will see that in the next part of the video.